So I want to thank Hudson Garrett, Dr. Garrett, for taking us through what I consider to be yet another one of the areas of his expertise and his extensive experience working not only in the healthcare side, but also the industry side and the value analysis side. So all of these portions coming together. So he's agreed to help us kind of undergo a primer about germicides. Where are we now, and what have we learned throughout the pandemic, and what's perhaps are some new approaches that we should be taking. So Hudson, can I turn this over to you?
Sure, absolutely. Well, thanks so much for having me, and good to see so many familiar faces. I hope everybody had a wonderful New Year as well. As Dr. Kirko mentioned, she asked me to talk about germicides, which is definitely a subject that I could go on for days about. I had the opportunity to sort of work on different sides of the house with this particular topic. I worked 10 and a half years as the VP of Clinical Affairs for PDI, which is actually at the time and still is the largest germicide manufacturer in the United States. And so it was an interesting experience, right? Because I worked on sort of the different clinical elements, worked a lot with clinical education, but also got to see the entire regulatory scheme of germicides and really disinfectants. And it was quite fascinating to see sort of how that operates. So for today, I want to make sure we sort of talk about disinfection in terms of healthcare associated infections and how it can help stop that. There's a lot of disconnect, I think, between sort of what we understand about environmental transmission and what we really don't understand. We'll talk a little bit about spotting classification and how that relates to environmental disinfection of medical devices and surfaces. We'll speak about regulatory aspects of disinfectant use and sort of where that lives within the federal government. And then we'll finish up a little bit with the disinfectants themselves and how to address some of the challenges with those different issues, right? With labeling and understanding claims and where to go for the most accurate source of information. And finally, we'd be kind of remiss if we didn't address the pandemic applications of how we need to sort of be ready for that next big one, if you will. So our goal with the environment of care, though, is pretty simple, right? It is not to get to a germ-free zone. We don't want to live in a world with no germs. We know that microorganisms do play a vital role in protecting us from an immunity standpoint. They certainly live in the environment. They colonize the environment. There's that microbiome that we have ourselves, but there's also a microbiome in the environment itself. And so this is present in healthcare, right? And we see this across different healthcare settings quite routinely. And so I always sort of laugh when somebody says, well, my operating room is sterile, as an example. All right, there's no operating room that's sterile. We may bring sterile supplies into the operating room. We may handle it aseptically. We may try to maintain sterile technique, but there's no operating room anywhere in the world that is truly sterile. We all know that that's simply not possible. So our goal is to sort of fragment out the dangerous microorganisms that will potentially cause either our patients or frankly also us harm, right? And get rid of those. Now, if we sort of overlay this with what we're all familiar with, that chain of infection transmission, we know that we've got to have a bad bug, right? We've got to have a route of transmission, and we've got to have that susceptible host. And so what we're trying to do, right, when we think about that contact transmission, where maybe we're going to touch that contaminated surface and then touch another surface, or touch that contaminated surface or patient and then touch another patient or another surface, we're trying to break that, right? And there's multiple different ways that we can do that. First and foremost, of course, is our basic hand hygiene. But there is a very close second, which is environmental cleaning and disinfection. And we'll talk about the differences between those here in just a second. But our goal should be to stop the organism from moving through this process so that it never reaches that susceptible host, whether that's a patient or certainly it could also be us. And so I break this down into sort of three different buckets of where healthcare associated infections come from. The contaminated hands, right, of the healthcare workers or the patients themselves. Certainly there's the contaminated clinical environment of care, which is what we're talking about today in terms of germicides. But then there's also the patient's skin, which we break quite frequently. Once it's no longer intact, we have lost that natural barrier for infection prevention. And so our goal today is to really apply germicides and disinfectants, right, across the continuum of care so that we can leverage these to stop transmission of environmentally challenging organisms. And there's really two different ways to think about infection control, right? There's the way that we're going to react to the situation, which, we're already behind the 8 ball. We've already got an infection. We're dealing with the transmission. We're dealing with the outbreak. Or we can be on the total left side of this equation, which is more proactive. And so germicides and disinfectants and cleaning agents, whether they come in a form of a wipe or a tablet or spray, it really doesn't matter. We're trying to prevent the microorganism from ever causing contamination. And that is going to give us a lot better of an advantage, right? A home field advantage, if you will, to prevent transmission. Again, once we get on the right side of this equation, we're unbalanced. We're unable to stop that as quickly as possible, and we're going to expend a lot more resources, which is going to make this much more difficult. And there's a lot of really hot topics related to disinfection that we frequently hear, and also some misinformation. For example, we hear the term one step, right? One step essentially means that you can use that disinfectant product to both clean and disinfect without doing a second step, without using a second wipe as an example. And what we found is that most environmental surfaces are typically soiled. A lot of the ones, especially in our clinical environments, they have some type of visible soil. And so a one-step product or a one-step approach is really not appropriate, right? If you look at the CDC guidelines as well as other guidelines and regulations out there, they talk about the value of really separating these two tasks into two different unique steps. Cleaning is simple, the removal of bioburden or soil through mechanical friction, right? We do that in our house all the time. Even something like dusting is a form of cleaning. Whereas disinfection is actually where we kill those microorganisms. And so this is a very, very different thing. Now, let's talk about this in terms of these commercially available one-step products. There are many, many disinfectant products on the US market that say they're one step, and they are, right? They can be used for a clean environmental surface with one single application, right? But that means that there's no visible, residue or soil or anything. But again, the majority of our surfaces are going to have something on it, some type of, body fluid or some type of contaminant. And so we want to think about that. There's also new formulations, which we'll go through here in just a minute. And there's some ones out there that have got a lot of promise, especially as we get the formulations more stabilized. And there's ones that can be a little bit more challenging, things like phenolics, right, which seem to be making a little bit more of a comeback here in the last few years, which is quite interesting given some of the potential health hazards that are associated with that particular type of formulation. Now, if we use a germicide, we've got to make sure it works. And there's a couple of different ways, from the really, really cost effective all the way through the more, I would say costly approach to that. And then lastly, we've got to understand, as Dr. Carrico mentioned, sort of the value analysis perspective. How do we evaluate these products? How do we make sure we're selecting the right thing? How do we make sure the staff is appropriately trained so that not only are they using it correctly, but frankly, that they're safe in the use of this? And really looking at this from a pandemic perspective. So there's lots of different movement that's happened really over the last, let's just call it 30 to 40 years in terms of disinfection, right? The majority of the US market is still made up of really two basic chemical compounds. One is chlorine-based, which is our bleach-based, and the other is going to really be a quaternary ammonium and an alcohol-based. And so quaternary ammoniums, just for familiarity, are found in laundry detergents and many other chemicals. They're highly effective disinfectants, but they do require typically a little bit more of a contact time. So you'll see some of the products on the market do have some type of alcohol sort of co-ingredient, if you will, that allows the solution to have a faster overall contact time, right? You'll also see some solutions out there that are high concentration alcohol. A lot of these were really designed for things like computers, particularly telemetry screens and touch screens, which are a little bit more sensitive to, certainly solutions like bleach or even some of the quaternary ammoniums. And so these solutions can be used there, but a lot of them are not approved as disinfectants. And so it's important to know what you're using and are you using it for its intended purpose. You also see, accelerated hydrogen peroxide. Until very, very recently, that was probably the hottest one on the market for really the last 5 to 7 years. And there's different concentrations of this, different parts per million, lots of different evidence that hydrogen peroxide is a very effective chemical to use, but it does have a unique sort of shelf life. It also can potentially have an odor. And so there's a lot of stabilizers and sort of formulation additives that have to go into that to make sure that it's going to be really well received within a healthcare facility. One of the ones that is really hot right now is hypochlorous acid. This is one that has some similarities to bleach solutions. It's got a lower pH. What's interesting about this is that it has essentially all of the properties, if you will, disinfectant-wise, of bleach solution. So it's got a really broad representation of activity against microorganisms, but it really sort of degrades down into water. The challenge with it is stability. And so you'll see some formulations coming out, some have already come out. There's a few that are coming out in the future, as I'm aware, that are in this category. And so the reason I share this is that while there are lots of novel things that come out, there's also a lot of mainstays that have great efficacy, a great history of safety, are widely available in a variety of different formulations as well as delivery systems. And so we need to think carefully about what are we using and where do we spend our dollars, right? Because we all know that dollars in healthcare right now are extremely tight. The last one that I mentioned there is antimicrobial environmental surfaces. And I'll be honest with you, I've got very mixed emotions about this. If you go out and tell environmental surfaces that, these surfaces are coated and they should resist growth of microorganisms, what do you think is going to happen in terms of cleaning of those surfaces? I can pretty much guarantee you that cleaning of those surfaces is going to go way down, right? Compliance with our protocols is going to go way down because they're under the false assumption that they don't need to focus on those areas. And so, yes, there are some interesting technologies out there, but again, I have to say from a value standpoint, is that where I want to spend my dollars? Or do I want to spend it on something that's a little bit more robust? One of the other things too is to really think about application contact time and persistence on surfaces. And so this is just one example that was in the literature back in 2020 that looked at the SARS-CoV-2 virus that causes COVID, right? And so you can see a wide variety of persistent timeframes that it can survive on different types of surfaces, right? Things from aluminum to rubber to Teflon. And so this tells us that we have a pretty big group of parameters that is both high and low that we've got to be aware of. And so does this really relate directly to disinfection? Well, it doesn't change the way we use the disinfectant, but it certainly reminds us of the importance of that, right? Now, just because something lives on an environmental surface doesn't mean it's going to jump off the surface and get into the patient or into us. That's not how the organism works, but there is definitely a likelihood of transmission, especially in our very tight spaces in which we work. Increased length of stay is certainly going to change that as well. How many times are we entering the room? What types of things are we doing? How much equipment is in there? All of that is going to play a role. And so when we think about overall contact time, what we mean by that is the longest amount of time it takes for that germicide to actually kill all of the organisms on the product's label. So let me give you an example. So let's say that you have a bleach solution and it kills all bacteria in 30 seconds, but it takes another minute to kill viruses, and it takes 3 minutes to kill fungal organisms, and it takes 5 minutes to kill mycobacterium, right? The overall contact time is 5 minutes. But you'll see many manufacturers out there that will say, oh, it's a 30 second or a 1 minute contact time is the most common. And in reality, that's really not the case, right? And so this is something that we want to make sure that we're looking very carefully and not relying upon the manufacturer to dictate to us what the true contact time is. As infection prevention experts, it's up to us to say, this is the contact time. I've reviewed the label. I'm not relying simply upon an industry representative to tell me what that is. And one of the other things to consider too is when we say something's clean, does that really mean it's clean? Now, this is an older study that was done back almost 20 years ago that looked at the presence of VRE in a clean ICU room. And the picture is pretty bad because it's old, but you'll just notice that all of the green X's represent a site that VRE was recovered in this clean room, right? And so it tells us that environmental transmission and colonization of the environment is really common, right? And it also reminds us that we have to give environmental services both the training as well as the time necessary to properly turn these rooms over, right? We can't rush them. In a room like this, 10, 15 minutes is not adequate to properly disinfect this room and make sure that the next patient that comes in is going to have the most, reduced possibility of transmission. And as we mentioned earlier, we talked about the differences with cleaning. What we find with germicides, right, or disinfectants, is that they really come in sort of two different forms. There's low-level disinfectants and there's intermediate-level disinfectants. Low-level disinfectants are certainly the core minimum requirement in the CDC guidelines. So you can check the box if you're using low-level disinfection, you're meeting the CDC requirements. However, the majority of the market, rightfully so, has moved to an intermediate level disinfectant sort of principle. And the difference between these two is that intermediate level disinfectants are effective against mycobacterium, right? The most common example that we're all familiar with mycobacterium is Mycobacterium tuberculosis, otherwise known as TB. Now you may say, well, what the heck did he just talk about TB? Because that's airborne. Well, we're not worried about, environmental transmission. But Microbacterium as a class of organisms on environmental surfaces is extremely resistant, right? It's really hard to get rid of. And so the majority of disinfectants, if they're not properly formulated, they don't have the right active ingredient, the right inerts, et cetera, and the right contact time, it's not going to actually kill that. I've seen hundreds of tests fail with really good disinfectants that cannot pass the intermediate level disinfectant test. Now, another reason that you may want to think about this is that back in the 80s, when HIV hit, OSHA put out that memo, right? And some of you may remember this if you're old enough to remember. And they basically said you need to use a 1 to 10 dilution of bleach for blood spills, right? And the reason behind that was very similar to the intermediate level disinfectant argument, was that we have, a potential dangerous organism that we need to successfully inactivate. Now, luckily, right, HIV is extremely easy to kill on environmental surfaces. It doesn't take much at all. You can really pour rubbing alcohol on it and it's dead. But our more resistant pathogens that we have in today's environment definitely take a more concentrated effort. And so the use of an intermediate level disinfectant is going to give you that microbacterium claim. And the thought process is that it also takes care of the things that are less resistant in sort of that pyramid of microorganisms. And so that's why you see a lot of folks that are doing that. To check and see if your germicide has that claim, you can look on either the product itself, but I will tell you a lot of manufacturers are leaving that off because that tends to be the longest contact time. But the absolute best place to go is the EPA's master label, which we'll talk about here in just a moment. So as we think about Spalding's classification, right, we're falling in terms of germicide use into the non-critical category. So we're talking about stuff that's not going to be used on non-intact skin. So, blood pressure cuff, pulse oximeter, stethoscopes, all of that type of stuff. And this is the biggest bucket, right? So if you look at all the supplies used and all the equipment used in healthcare, by far, this is the biggest bucket used. Certainly right behind that is going to be the sterilization market, where we've got lots of instrumentation. And then behind that would be semi-critical items. So from a regulatory perspective, it can get a little bit confusing, right? The EPA owns all germicides. And so if it is used on an environmental surface, it is going to be owned under the EPA side of the house. And this includes things like electrostatic machines and foggers. The FDA on the other side, the other hand, deals with anything that comes in contact with patient skin. And so if you have high-level disinfectants or sterilizers, those are going to be owned under the FDA side of the house. Right. And so what's challenging is that currently germicides that are used in sprays and wipes and liquids are heavily regulated by the EPA. But you'll find that things like UV and electrostatics are not currently regulated by the EPA, right? A little bit surprising, and they still have not done this, which is interesting, but they are not currently regulated. And so there's a lot more disparity in what you'll hear about these devices in the market as an infection preventionist than what you'll see for some of the other more common items like disinfectants that have been out on the market for years and have very, very tight regulations as to what they can do. Now, one thing that you want to know for sure is, and this is part of our regulation piece, is that if they're using any disinfectant in the US, it is required to be registered with the EPA, right? And this is really under the antimicrobial pesticide group. This is also in the same category as what they spray out of planes on our crops, right, to keep bugs away. It's the same different classification. The difference is these are used in healthcare settings, right? One of the other things to do is to consider that you need to make sure you're aware of what personal protective equipment is required, right? And sometimes you'll get information that's really complete from our partners in industry, and other times, frankly, you won't. And so it is your responsibility, it is your facility's responsibility to make sure you're vetting these products appropriately and taking all necessary steps. And so, as we mentioned before, the EPA holds the power in terms of germicides, right? And so there is something you want to be aware of called FIFRA. It stands for the Federal Insecticide, Fungicide, and Rodenticide Act. Federal Insecticide, Fungicide, and Rodenticide Act. Now, that sounds like a real whammy of a tongue twister, but it's really a fancy regulation that says one thing. You can't use anything that's regulated by the EPA off-label, right? And that includes everything from disinfectants to hairsprays, which are also regulated because they're actually under pressure. So anything that impacts the environment is going to have some type of EPA regulation around that. But FIFRA is very specific. You can't use something that's off-label. So that means contact times, doing a dilution for which it's not approved, adding stuff to it, anything like that, you will be liable as a facility if you do that. One of the best resources that's available out there on the EPA's website is their label product information system. This allows you to type in the manufacturer, the product's name, or the EPA registration number, and instantaneously get all of the documentation that they're approved to promote, right? It includes things like contact times, their master label, their safety data sheet. All of that type of information is available through that public information resource. And as I mentioned before, FIFRA is not something that you want the government to come after you for. There are penalties. These are fines that can be quite large. I think the largest that I've seen was about $25,000. That was something related to a pretty big mishap where somebody deliberately did something. And I'll tell you that compared to places like OSHA, EPA does not leverage fines in this area very often. And so it really requires a complaint to come in or some type of adverse event. A lot of times Joint Commission may notify as an example, or CMS may notify, but there is a formal complaint process that takes place. And EPA does have regional offices across the entire United States that handles that. One resource that I'll send to Dave to have posted with the Grand Rounds website is this, and I call this the Bible, right, of disinfection. And it's really called the Label Review Manual, right? And in this manual, you see it has 18 total different chapters. Don't worry, you don't need to review all of these, but there are some fantastic resources in here that as infection prevention experts, we need to be familiar with. Things like general labeling requirements and, what are the precautionary statements in terms of the use of PPE? How do I make sure that I'm aware of workplace protection and directions for use, right? What about, the different types of labeling requirements? All of that is contained here. So you can go and download this for free. I'll give Dave the website link, or he can drop it in the chat, which he typically does. And this is something that is a fantastic resource for you to have. It also allows you to call BS when people are giving you information that is just not accurate, right? Again, we can't rely upon others to dictate our clinical practice. We must do our own research in order to protect ourselves. There's also free training available if you're interested. I'm not going to lie and tell you that it's the most exciting, right? But it is thorough. And so if you're looking for a better understanding at a granular level of the regulatory scheme of this area, which I strongly recommend that you do, I've included the website there, and you can certainly use that at your leisure. They do a nice job of being thorough, but like I said, it's not going to be the most riveting training that you've ever participated in, but it is definitely going to give you a good broad overview of this topic. One of the other agencies that we've got to be worried about with germicide use is OSHA, right? OSHA has one single purpose, which is to protect the worker. They have no interest in protecting the employer. And so there are multiple different places where germicides are mentioned in OSHA standards, most notably the Bloodborne Pathogens Act, right? And so this is something that you'll see in there that is out there, available on the OSHA.gov website. There's also that ETS that is still sort of in limbo. My gut tells me OSHA's not coming out with it, but who knows what will actually happen in the future. But OSHA carries a big stick, right? You don't want OSHA coming to visit you. I've lived through 2 OSHA inspections. Both of them were very not pleasant, and they also had pretty big fines, right? And OSHA has a lot of ability to leverage fines. They can also actually shut down facilities through the court process and get an injunction. So definitely something we want to be aware of. So how do we make sure we select the most appropriate germicide? Well, first of all, as we mentioned before, it's got to be EPA registered, right? You'll hear this term hospital grade. Hospital grade, I don't want you to worry about because it's really an antiquated term. When EPA came up with this, they were basically trying to say, we need something that differentiates old disinfectants, right, from the new ones that are specifically designed for use at that time in hospitals, right? So this was many years ago. We recognize now that disinfectants are used across the entire continuum of care. And so it's a very, very antiquated term that essentially represented that product at the time was effective against 3 microorganisms that were deemed important in healthcare, right? Specifically in hospitals at the time. And so it really doesn't carry a lot of relevance now. Broad spectrum, though, does. And we'll talk about what that means here in just a minute. Look for an intermediate level disinfectant if you can find one, right? Within low overall contact time. Now, when I say low, I'm really talking about 3 minutes or less. And there are many, many products out there that can help you meet that criteria. right? We don't want it to tear up our equipment, so we've got to have good equipment compatibility, and it's got to be safe for our patients or our residents if we're in long-term care, and our healthcare providers from a safety profile standpoint. So I look at three simple things. It's not very hard, right? First and foremost, does the germicide work? Does it actually take care of the microorganisms that I'm trying to target? And if the answer is no, then I don't proceed to step two, right? But if the answer is yes, then I'm going to say, is it going to be safe, right? Is it safe for my entire team, the patient, the resident, the healthcare team, the environment itself? And if the answer to that is yes, then I proceed to my final leg of my stool, which is it compatible? Is it going to be okay to use on the many different pieces of expensive medical equipment that I have? And I don't want it to degrade that or void a warranty. And this can be really challenging. One thing that I would encourage you to do from a compatibility standpoint with your germicides is pull the instructions for use for the medical devices that you have. You can work with your biomed department and figure out what is approved for use. You'll find that many of them either have very specific products or they're much more broad. I'm really a fan of the much more broad because that gives you a lot of leeway. If they say a 1 to 10 dilution of bleach, well, that can be a lot of different products versus it has to be this particular product or it has to be that particular brand, right? That can make it a little bit more challenging. And then I need to think about a couple questions, right? Who's going to use it? Where are they going to use it? Where are they going to store it? How often will it be used, right? What training do I need to have in order to be safe and reliable in the use of these types of products? Because you don't want someone misusing it. And you also don't want to have a failure in your infection control process. Now, as I mentioned a minute ago, broad spectrum means different things to different people. So this is a really good, I think, definition to sort of memorialize, if you will. So we're looking at gram-positive and gram-negative bacteria, your enveloped and your non-enveloped viruses, certainly your multi-drug resistant organisms, pathogenic fungal organisms, things like Candida, right? You've got your bloodborne pathogens, which are required by federal law. And then you've got this sort of bucket for emerging pathogens. Well, we just went through this, right? We had COVID, we've had, now we've got another outbreak of Ebola, right? So these emerging pathogens that are unsafe for laboratory personnel to test with disinfectants are going to be in this bucket. And EPA does have specific guidance out there that tells us exactly what we can do in terms of that. And so essentially, it's very similar to like the mycobacterium. So they'll say, okay, Ebola is this type of virus. What products are in a similar class? Meaning that if they've got a claim against a very similar virus, and then if they do, they essentially grandfather them and say, okay, this list, if you will, is effective against Ebola. Doesn't mean that they tested it against Ebola. It just means that they know from a scientific standpoint that the performance of the germicide is going to be effective against a similar microorganism based on its structure. From a microbe standpoint. So ingredients themselves also matter, right? This can be a little bit hard to get. The active ingredients are required by law to be listed on the product. So that'll typically be listed directly in the product packaging. However, you will typically not see the inactives or what we call inert ingredients, right? The biggest inert ingredient typically is going to be water. Right now, that can make a difference because if you are using non-filtered water, for example, this can be problematic because your disinfectant may become contaminated, especially if you have a failure in your disinfectant active. The other thing to think about too is, as I mentioned before, equipment compatibility, right? How do we make sure that it's going to be safe for our equipment long-term with repeated use? And one of the biggest areas I think that's a gap is repeated use toxicology. Right. And I've dealt with both OSHA and a couple of different state EPA offices on this, and also dealt with NIOSH, where they're very, very interested in what's the exposure risk to us as healthcare workers when we're using something 100 times a day, right? Which is very, very common for somebody like environmental services. We need to make sure that with repeated use, there's not going to be any contact dermatitis or any respiratory issues or asthma or anything of that nature. And there is some data in the NIOSH database that does track specifically things like respiratory illnesses. And so it's important for us to be aware of that and know what we're looking for to certainly do a very deep occupational health investigation if we have any employees that present that way, and to really do a formal investigation. We know that environmental services can either be in-house or contracted. And so this is becoming more and more common, especially in places like long-term care, which is the example that I included here, where if you've got in-house, right, you may have more direct insight or oversight, if you will. And if it's contracted, it can be a really big, hodgepodge, right? And so that's going to be a challenge. Now, how do we actually make sure that we're going to be successful in our efforts? Well, we can do different forms of environmental monitoring. Now, the cheap and free way, right, is to do visual observation, where I'm going to look, I'm going to check, I'm going to look and make sure that I'm doing leadership by rounding. We can do things like bioluminescence, which, cost a couple dollars, if you will, where we're going to mark surfaces, see with the black light if they actually clean those, what their technique looks like. Again, a great tool for process monitoring. There's ATP monitoring, which looks for the presence of adenosine triphosphate, which is going to tell you if there was recently dead life or currently living life. Doesn't tell us what it is, but it just tells us with a quantifiable number what is currently present on the surface. And so it gives us a good idea of how clean that surface is. And I'll tell you, there's lots of cool technologies that are coming out. Some of the research and development was a little bit delayed, I think, over COVID, but there are things that are coming out that help us. I've even seen wipes now, for example, that actually, show you visually with a color where you're actually, using that on the surface. And there's some mixed, I think, reviews with that for sure. So what required training do you need to do in terms of germicides? Well, first and foremost, there's a sort of cadence that you need to follow. And it's at the time that you hire the individual, certainly annually, and anytime you change a product or a process. So if I'm going to, if I, for example, am a Clorox house or PDI house or a Metrix house, if that's my product category of use, then I need to train my people as soon as I hire them. I need to retrain them annually, or if I change a product. And many of these large companies are coming out with new stuff every year. And so it is very likely that you may make product shifts at that point. And you want to include basic things like the PPE and indications for use, and what happens if I get it in my eyes is a very common one. So they need to understand the safety data sheet. And from an environmental standpoint, certain states do have very, very strict parameters, California being one of them, where you cannot wash some of these things down the sink or toilets or anything like that. So they want you to do a specific type of waste disposal for any residue left in there. Now, from a germicide standpoint, it really fits into our core category of infection control measures, right? Because we're going to do that good cleaning and disinfection on a daily basis. And certainly anytime something is visually soiled, right? And that gives us that more proactive approach to environmental hygiene. Now, there's other things like electrostatics and UV and foggers, right? That can be used on an adjunctive basis, but they really are not to be used until you have a good grasp on the basic principles, which are cleaning and disinfection. And so while there's lots of cool things coming out there, like far UV and all kinds of new technologies, right? They don't replace the need for our mechanical friction with cleaning and disinfection. That really is going to continue to be the mainstay, in my opinion. And so a couple of questions before we get to any questions that you have. First and foremost, when I'm looking at germicide product selection from a value standpoint, right? Does it work? Is it going to meet my unique facility requirements clinically, right? Do I have a reasonable contact time? Is it under 3 minutes, as I mentioned before, right? Is it going to be safe for my patients, my residents, and my staff? And then what technologies, right, should be used? Are there some new things out there that we might have? Now, I'm a big proponent of looking at new technologies. I like to look at innovation, but there is always a risk, right? And so do your homework. Make sure you're checking to make sure this is something that is already EPA registered. You do not want to be caught breaking federal law by having a disinfectant that's not yet registered. That can get you and that manufacturer into a whole host of issues legally to do that. Think about the room clearance procedure as well. How quickly can I go back into the room? This is important for things like foggers or misters or electrostatics. Some of them may require a 10-minute dwell time where you can't re-enter the room. Obviously, many of these may require you to leave the room altogether. And if it's a double occupancy room, you can't have another patient. And so that's something that we need to be aware of also. So finally, how do we prepare for the future? Right? Well, CDC put out this really great thing during COVID that talked about sort of different levels of PPE. And I sort of took this and said, okay, let's make this into something related to disinfectants. And so if we use the same sort of stoplight approach of conventional, contingency, and crisis, right? Conventional is everything is perfect. We're getting our normal products. There's no back orders. There's no substitutions. We've got ready to use. Everything is peachy, right? But then we've got contingency where We certainly operate frequently. This happens with just supply chain and just frankly with everything. Now it seems where we have these we've got these substitutions and we've got different formats that come in, right? And so that can be problematic for us. And then when we get into the crisis mode, which unfortunately at some point we got to this place during COVID, right? This is where we're looking at completely different products and completely different formats and delivery systems. And so there are different ways to deliver germicides. The ready-to-use wipes, sprays, and liquids are the most common, right? That's going to fall in your conventional category. You may have to look at alternative ready-to-use products, most often typically in the sprays and liquids, because once you have a problem with supply chain, the wipes go out quick, right? They're not available. But in a crisis mode, and also from an innovation standpoint, you're seeing more and more of these sort of dilutable liquids and dilutable tablets and the concentrates and the things that have a little bit more of a broader spectrum in terms of application, and they don't require you to have a lot of stock. You can do the dilution yourself. Now, of course, when we mention the word dilution, we should all be worried about who's going to dilute and are they properly trained and am I going to have efficacy, right? Because that is always a potential risk. And that's why it does take an entire team, right? It really requires us, not just environmental services, not just infection prevention, not just biomed, but really everybody to be a part of reaching sort of the end goal, which is a clean and sanitary environment with the bad organisms gone, the good organisms still there, and a safe environment for us as well as for our patients. So as we summarize, right, disinfection and the use of germicides continues to be a big part of this process, right? This is not something that's going away. We've had disinfectants out there for 30 plus years. Have there been innovations? Absolutely. But have our lessons learned and our fundamental principles changed a lot? Not really, right? We need to sort of rely upon what we know works, and that's good attention to detail, good friction, good mechanical efforts, using a proper product, and knowing what you're doing. And that all goes back to training and competency. Training alone does not equate to competency, as we all know. You can be really well trained and really incompetent. We see that every day across healthcare. And a lot of this is going to be, as Dr. Carrico mentioned, based on a formal value analysis process. I've had the fortune to work with that group over the last 6 months pretty heavily. And it's really interesting to see sometimes the disconnects between different professions and the silos that we exist in. And infection prevention is included in that. And so that's why it's great to bring different disciplines together in this format of grand rounds, where we've got pharmacists and advanced practice nurses and public health professionals and infection preventionists and everybody under the sun. But we know that our internal partner in this fight is environmental services. And so we want to make sure we're partnering with them. So with that, I'll stop and see what questions we have and turn it back over to Dr. Carrico.
Thanks very much. I found this so good, because as we dealt with COVID and the infection prevention and control side, we confronted exactly what you said. Not only our usual chemicals were unavailable, but our usual ways of applying those chemicals were unavailable. So we went through situations such as make your own wipes, that type of approach, to having a number of products marketed directly to infection preventionists that were either not yet FDA approved or had FDA approval, but perhaps not the product that we needed in order to address, the organism. So, giving us that checklist almost, that ESC, that looking at the efficacy and so forth, and having a plan. And I know that where a lot of facilities now seem to be using the combination alcohol quat. I see that more and more as I'm going. So some of you talk about some of the pros and cons. If just because I think that is becoming so prevalent, any particular comments, again, just to kind of reiterate for that type of product?
Yeah, so alcohol and quats have been around forever. They've been around for 20 plus years. There's different levels of quat and different levels of alcohol. Keep in mind that the higher the alcohol content, the faster the contact time will be, right? So from an efficacy standpoint, as far as getting activity quick, it works great. The higher the alcohol content, also typically the more drying it is to the surface that it's going to be touching. And so when you're using isopropyl alcohol, which is what's used, that's different than hand hygiene where we use ethyl alcohol. Isopropyl, it can be particularly drying. So we need to think about equipment compatibility. And so what I would encourage anybody to do is to do two things. One is to look at the efficacy and say, what's the advantage of this formulation together? Sort of what synergistically does it do that's different? And I think secondarily to that is, let me know what equipment compatibility studies you have, right? If you can find a broad representation of things like stainless steel and the common plastics used in IV pumps, which is a really common one, and it passes all of that, then you should be good, right? That should be a pretty good place, a good sweet spot for you to be. But as soon as you start to see some of those things fall apart, you know that you've got an unbalanced equation, right, of sort of the safety and the efficacy and the compatibility. And so you can see great products. I remember about 11 years ago specifically, there was a product that a manufacturer was going to introduce. They spent millions of dollars. And I'm sure you remember which one I'm talking about, Dr. Carrico, but this is a massive company that we were all familiar with. It's now merged, so it doesn't matter at this point. But they spent millions of dollars to launch this at APIC, and infection preventionists spent 5 seconds with it, and it smelled so bad that they never brought it to market. It had great efficacy, it had terrible compatibility, and it had a terrible smell. And so the thought of bringing something like that into the market where it would be offensive as a scent, they never considered it. And so I think our job is to look at the bigger picture to make sure that our patients will be satisfied, our teams will be happy. And just like we talked about in the quadruple aim, it's not just about the patients anymore. It is absolutely about us. The CEO's number one concern is staffing, which means we need to retain staff. We don't need to do anything to alienate our staff or make them feel unsafe at work. And so giving them good disinfectant products that are going to be neutral smells and really good for the environment and have great efficacy is what people are looking for.
Yeah, and I think, part of that analysis, you mentioned about, who should be involved in product selection. And it's certainly the user, as you mentioned, because, the person that's going to be using the product will tell us either directly or indirectly through their action how likely they are to use it and use it correctly. So some strategies that maybe you have encountered through your work with our value analysis colleagues, that when you were looking, or if you were looking at a new germicide or a new approach to disinfection, what might be some partners that the infection preventionists should reach out to so that they've got a good group to look at that product?
I would actually argue that infection prevention doesn't need to own this. This really needs to be owned by environmental services. I mean, environmental services are the users. It's no different than infection prevention doesn't need to own IV start kits. Infection preventionists don't start IVs. Vascular access does, and the nurses do, and other healthcare professionals do. And so we need to let the area live with who does it. And so environmental services is definitely, they also own the environment of care standard in the facility. And so I think first and foremost is be a consultant, be a resource, be a champion to them, be a clinical support liaison to them to help them understand any science related to that from a micro standpoint. But they're really the implementation professionals at this. They're the ones that have to operationalize it. And so I think go there. I would also encourage you to certainly look, as you mentioned, at healthcare value analysis. A lot of those folks are clinical by background. Biomedical engineering is a huge player in this in terms of equipment compatibility. And then I think the other piece too is to talk to healthcare risk management. That is a group that has a lot of data about areas of risk throughout your facilities. And while they may not be directly related to disinfection, you will quickly find when you start to have conversations with an experienced risk manager that they will point out areas where we can leverage disinfection more appropriately and hopefully reduce the overall risk profile to our staff members as well as to our patients. And so I think get those people to the table and you've got a really good party that you can have and talk about this exciting subject.
Excellent. Do you think, I know we've talked on some of the programs about ECRI, and do you have some ideas about maybe some reviews or general information, and maybe share with the group a little bit about how they might find out that information from their facility in accessing that membership?
Yeah, so typically ECRI is, many healthcare facilities subscribe to it, and it has comparison tools that are very, very independent. They don't work with the manufacturers at all. They do a completely independent research, and they do cross comparisons. So if you're looking for the best bleach wipe or the best this wipe or the best disinfectant in general, you can go through the ECRI Institute and they actually have resources. And so what you have to find out is who owns that relationship within your facility. I know each hospital that can be slightly different, but a lot of times purchasing has access to that. So if you go to your materials management or your value analysis team, a lot of times they actually have a login. And so they can pull that information for you if you tell them what you're looking for. And it is a fantastic resource. The other resource that I would mention too is AVAP, which is the Association for Healthcare Value Analysis Professionals. That is a group that infection preventionists can absolutely join. It's a pretty cheap membership. And you can then download their value analysis tools and then use those to help vet some of these things, to really provide that clinical infection prevention input and be a better partner to this team where, the stuff that we don't want coming in the door, we need to stop it. The only way we can do that is, as you mentioned, to be sitting at the table actively as a partner and a resource to that team.
Well, I thank you so much. I know I could ask you questions all day, so I appreciate this opportunity. And I think a lot of this information has been not only foundational for us, but you did it in a way that helps us figure out what do we do with next steps. So I thank you very much. Dave, I see you have put up the closing poll. I appreciate that. And there is some information, additional information in the chat that can help take you not only to the recordings of these grand rounds, but to the infection prevention training site, and also some information about our upcoming grand rounds. So I hope everyone has a good rest of their Wednesday, and thank you much. Thank you again, Dr. Garrett. Wonderful as always. Appreciate it.
Thanks a lot. Take care.